Okay. Howdy, our names are Cassandra Fernandez and Alondra Curiel, and today we are going to present our biochemical test lab part 3. Introduction. This lab focuses on identifying bacteria through biochemical tests that reveal their enzymatic activities and antibiotic resistance. The catalyst test detects bubbling from the breakdown of hydrogen peroxide, while the oxidase test identifies bacteria with oxidase through rapid color change. The nitrate reduction test determines if bacteria can reduce nitrate to nitrate or nitric gas, and the urease test detects ammonia production from urea hydrolysis. Finally, the Kirby or antibiotic susceptibility test measures bacterial resistance or sensitivity to antibiotics. Together, these tests help distinguish bacterial species. Materials. For the materials, we used urate tubes, nitrate broth tubes, petri dishes, agar plates for antibiotic discs, sterile cotton swab, sterile loop and needles, a Bunsen burner, microscope, slides used for catalase, and for the reagents, we used hydrogen peroxide oxidase reagent, which is a tetramethyl P phenylenediamine, nitrate reagents A and B, uromedium, pre-prepared urobroth tube with phenol red, antibiotic discs, which were the bactricin, no, novobiocin, and optocin. And then for the culture, it was the Aphyllus E. coli, P. aeruginosa, S. aureus, S. epidermidis, Staphylococcus, um, Saprophyticus, S. pyogenes, S. agalitae, and unknown 1 and unknown 2. For the procedure, we start off with the catalase test by placing a small colony on a glass slide and adding a drop of 3% hydrogen peroxide, and bubbling indicates a positive result. Meanwhile, if no bubbling appeared, it indicates a negative result. And then oxidase test, we swabbed a colony with a sterile cotton swab and then added 2 to 3 drops of oxidase reagent and observed for rapid color change, which was indicated as purple, and it was supposed to be positive. It was if it was indicated as purple. And then for the nitrate test, we inoculated nitrate broth with a durum tube and incubated. Then we added the reagents A and B, and then red color indicates nitrate reduction, and if no color was visible, we added zinc red, and after zinc, it is negative, and no color is positive. And then for the urease test, we inoculated ure broth and incubated, and then a pink color shows urease activity, which indicates positive. And then for the antibiotic susceptibility test, the we swab cultured evenly on an agar plate, then placed antibiotic discs and incubated them and measured zones of inhibition to determine sensitivity or resistance.
For the catalase results, in the first one, SRS, we saw immediate bubbling and we interpreted that as a catalase positive, which is expected for Staphylococcus. For our unknown 294, we saw no visible bubbling, which is a catalase negative, which would be consistent with Streptococcus or Enterococcus or any other catalase negative taxa. For unknown 114, we saw immediate bubbling, which is a catalase positive. This helps rule out Streptococcus and Enterococcus because it bubbled. And for S pyogens, we saw no bubbling, so it's catalase negative, which is expected for Streptococci. In our oxidase tests, for E. coli, we saw no color change, which is just an oxidase negative control. For P. aeruginosa, we saw purple, a purple color change at the tip, which is an oxidative positive control. For unknown 114, we saw purple at the tip, oxidase positive. And for unknown 294, we saw no color change, which is oxidase negative. For the nitrate test, for unknown 114, we saw a red color change, which we interpreted as positive, which was nitrate to nitrate. For E. coli, we saw a red color change, which was a positive result, which just we interpret that as an okay control. A. facilis, we saw a clear but slight pink at the bottom. This just interpreted as negative, no nitrate reduction. For P. aeruginosa, there is a deep pinkish color. This could be a control anomaly. It's negative, and it's supposed to have no color change. And for unknown 294, we saw a red color change, which just meant positive nitrate to nitrate. For S. saprophyticus, the no bison, we saw a tiny zone around it, which is expected. And we classified that as resistance. For S. epidermidis, we saw a large zone with the no bison, so we interpret that as sensitive, and that was expected. For S. agilite, for the bacitracin, we saw a small zone around it, which was just classified as resistant, and that's expected as well. And for S. pyogens, for the bacitracin, we expected as sensitive because it had a little bit of a bigger result. So that's expected. And for both the S. agilite and S. pyogens, no biosin had no reaction because it's not really used for strep. Conclusion. In this lab, we performed catalase, oxidase, nitrate reduction, and antibiotic susceptibility tests to demonstrate how biochemical reactions help identify bacteria and distinguish between similar organisms. The control bacteria produced the expected reactions, confirming proper technique and accurate results. These methods also allowed us to narrow down the identities of our known samples based on their enzyme activity and antibiotic responses. Overall, this lab showed how biochemical testing is essential in microbiology for recognizing bacteria and understanding their clinical relevance. Thank you for listening to our lab video.